Welcome everyone to this installment of EREF's Science Sessions. We appreciate you being here. My name is Bryan Staley. I am president of the foundation, but more importantly, your moderator today for this science session, where we are going to explore PFAS in waste-derived compost. So just a few preliminary things before we get started here. For those of you who are not familiar with EREF, we are a nonprofit research foundation focused on funding scientific research to academic institutions, primarily providing scholarships to graduate students and conducting internal work on aggregating data and information, and then transferring that information to you in the form of an educational program, of which the science sessions that you're participating in today is a part of that. So the science sessions, if this is your first time here, welcome. They are a little different than a standard webinar. These are typically 45 minutes to 60 minutes. We're really just having a conversation with those who are in the know, our scientific experts, our researchers who are tackling a lot of the important issues that affect us in the solid waste, residuals management, diversion field. So these science sessions are really interactive. We encourage you to send along your questions over the course of the next 45 minutes or so, and we will try to get to each and every one of them, although that is not always possible. For those of you who are interested in a certificate of attendance for continuing education hours or whatever reason you might need that, you will get a link for that in Monday's follow-up email next week. Just a quick highlight, we are taking a little bit of a hiatus on our science sessions to insert a few training sessions, which will be more like a standard webinar. This is going to focus on electrical leak location methods around landfill liner systems, and there are 3 of them coming up in the near future here. We have one coming up later this month, and then a couple in June. And so if that interests you, I encourage you to keep an eye out for that, take a look, and certainly attend if that's something that is in your field. A quick thank you to our session sponsors, CEC, Golder, Republic Services, Waste Connections, Labella, and Enthalpy Analytical. Thank you all so much for helping us support these. We would not be able to offer these at such low cost without your help, and we greatly appreciate that. A few housekeeping items before we get going here. Everyone is muted. Please submit your questions throughout the presentation using the question button in your interface for GoToMeeting. We do have slides available for download. That's in the handout section of the platform as well. And as I mentioned earlier, everybody is going to get a certificate of attendance if you choose to have one. So, the last few things I wanted to mention before we get going here is that we have recently put up a PFAS resources webpage. Very excited about that. This is really a one-stop resource of most, if not all, of the research that is around the PFAS issue as it pertains to waste and other diversion endpoints and what's happening with PFAS in that. This is a curated list of scientific resources that you can go to. The curated list contains basically key resources that we have culled out of hundreds of different scientific publications that you can quickly look at. Or if you have an appetite for pain and suffering, you can download the Excel file that's on the same web page and access links to hundreds of these peer-reviewed reports. They'll take you right to the abstracts, and you can really get down and dirty in the details with what's going with PFAS there. The link is at the bottom of this slide. Again, that is also being shared in the handout, so you'll have that. And I encourage you to take a look at it. It is a resource, and it is freely available to you. So, to set up our conversation today, I wanted to just highlight a couple of things about how we understand currently PFAS to flow through society and the environment. So currently, when PFAS is manufactured, and again, it's manufactured, goes into hundreds of consumer products, goes into firefighting foam. Those consumer products ultimately are consumed, and packaging or other materials are thrown away, which puts it into the waste infrastructure, or we excrete it through our bodies, and it also ends up in the wastewater infrastructure that way, and that's how we see that entering into the environment. At some point, those materials either reach us directly or indirectly, and we receive some exposure to PFAS. So this is generally the framework that we tend to look through from a scientific perspective and trying to understand some of these questions about PFAS and about why it's important to discuss PFAS and what the potential implications are as we manage compost, as we manage recycling, as we manage waste that might go to landfill and so forth. This flow map, if you will, becomes rather important. So when we talk about this flow through society, then the next question is, all right, it ultimately gets to us. How do we get exposed? And there are really four primary ways. One of them being the more dominant way. I mean, we can breathe it in. A classic example would be carpet dust. If you have carpet in your house and it has been treated with a stain-resistant type of chemical, more often than not, that chemical would contain PFAS. And over time, as you walk through the carpet and so forth, you'll breathe that in. Dermal exposure is also been noted, although the science suggests that's not a primary actual exposure pathway. But certainly with treated textiles, clothing, sunscreen, those are things that you might that your skin might come into contact with, and that's another potential exposure pathway. The bottom right is through drinking water or groundwater exposure, and that has predominantly come through firefighting foams and some of the more, I'd say, point source exposures of PFAS. And then lastly, which some research has suggested that's the most dominant pathway of exposure, is by eating and ingesting the PFAS. And that is through food that has come into contact with PFAS. And many times this food that has PFAS has been exposed to some kind of packaging material that was treated with PFAS. So right now, there are about a little over a dozen to 2 dozen studies that have focused on this, and that this is sort of summarizing that science. It says that really the dominant exposure pathway right now, as we understand it, is through eating eating materials that contain PFAS. So how does this translate to composting, which is our topic of discussion today? So as part of the PFAS resource website that I shared with you a few moments ago, we've been tracking the science and trying to understand what do we know, what don't we know. And this is a sort of a diagram of where the science currently resides with compost. Specifically, the solid arrows suggest that there are multiple scientific resources available that help us understand what PFAS concentrations are to some degree. I will caveat that by saying there's a whole lot more we don't know than what we do know with PFAS right now, and particularly PFAS in compost. So even though there's a solid arrow there, that is not an indicator that we know everything. In many respects, really this represents just a few studies that exist, and where you see a dashed line that represents we know very little about about that particular area. So when compost is made, you typically would see emissions, gaseous emissions, or leachate coming from those piles or runoff, and then the compost itself. So much of the work has been done around PFAS in compost material. Certainly when compost is then used as a resource, it could go to crops, it could go to landscaping primarily, and then if it goes to crops, you could potentially eat that crop as corn or lettuce or something like that, or you could feed that crop to an animal, and then we would then eat those animals, and then that could be another way that we could potentially be exposed to PFAS. So I think the big question really is, how did PFAS get into compost in the first place? That's the burning issue, and we have an expert here to help us understand that a little bit better. Dr. Youn Choi is an analytical chemist in the Department of Agronomy at Purdue University. And prior to that, she was at the Colorado School of Mines as a postdoc fellow and spent a lot of work looking at firefighting foam. So, and as I mentioned earlier, firefighting foam is one of those key materials that contain a lot of PFAS. So she's spent a lot of work and a lot of time investing in this PFAS question, and really issued one of the groundbreaking papers on PFAS in compost a couple of years ago, which is partly what we're going to talk about today. So Dr. Choi, welcome. Thanks for joining us today.
Yep, thanks for a great introduction, and thanks for all the explanation about compostings.
Yes, absolutely. So I have a couple of questions just to start us off with. When we're talking about PFAS, give us a sense for PFAS in compost. And PFAS are known to be found throughout the environment. Why are we finding PFAS in compost?
I know it's boring but scary answer, but PFAS is everywhere. PFAS is ubiquitous, and there are many cases the ingredient of compost already have PFAS. So for example, food compost, depending on the ingredient, the compost can have organic fraction of municipal solid waste, and in many cases it has food contact material. The food contact material has a lot of PFAS to have a coating to prevent any absorption of food onto their container. So that could be one possible reason we have PFAS in compost when we have organic fraction of municipal solid waste in our compost. In case of agriculture byproducts, it has less chance itself has PFAS, but when animal has exposed to PFAS contaminated water or they are fed with PFAS contaminated food, their manure and bedding can have PFAS. In case of spent mushroom waste also can have some kind of PFAS, and because the basic material could have PFAS, biosolid, I don't think I need to explain more about why biosolid has a lot of PFAS.
So, so you said mushrooms. That's interesting. So maybe just ask more out of curiosity. So for mushrooms, we're seeing PFAS in mushrooms primarily because of kind of drill down on that again for me. Why are we seeing PFAS in mushrooms? That's interesting.
So it's not from mushroom industry or something else, but the bedding, the pit, or anything used for growing mushroom can be already contaminated with PFAS. So one example, there was a recently article about PFAS detected in pesticide, but it was not because of the pesticide, but the container was fluorinated. So the PFAS released into the pesticide, and the pesticide applied material was contaminated with PFAS.
I see. Okay, so we don't have to be too worried about mushrooms specifically, but it's what's happening to the area or the mushrooms themselves that's causing that.
Right.
Okay. Well, that's helpful to know, because I think that really gets into this question of feedstocks that go to compost materials. You know, from your response on the mushroom question, I mean, that would suggest that really when we're talking about the feedstocks, that would seem to be the primary concern is understanding how PFAS is getting into the feedstocks that would go to a compost operation. Is that a fair statement?
So first of all, the main resource would be feedstock, and there could be really low chance from composting procedure itself, but there was some possibility from leaching PFAS from fluorine treated fabric covers.
Okay. So now beyond the feedstock, you sort of touched on that a little bit though. So would there be any other sources of PFAS that would contribute to the concentrations in a finished compost product? So if there are other things that have high concentrations of PFAS, that's what you're saying, is that beyond the feedstock, you know, one would just need to be mindful of anything else that might have significant concentrations that would be mobile that could transfer into the compost. Is that what I'm hearing?
Yeah, so there is no direct study about the cover used for composting, but when somebody studied research about outdoor clothing, and when it was exposed a lot and weathered a lot, the leaching of PFAS from the fabric texture was increased from 5 to 20%. So okay. Yeah, the initial leaching concentration would be important, but also it could be another little possible source of leaching PFAS from the composting processing.
I see. Yeah, and you highlight an important topic of research right now, and that is, you know, how do the breakdown of materials that are known to contain PFAS ultimately result in the transport of those materials? I know that's a big question in the landfill sector right now, is that all of these materials are going into a landfill. You know, are they sequestered there, or over time, as you know, microbes attack it, do we see some leaching from some of these materials? And that would really be any of the waste materials. And you know, so the assumption is that the leaching would occur at differential rates based on what the kinds of material that the PFAS is in, as well as what kind of PFAS it is as well. So those are a couple of key questions that need further exploration, most certainly. And I didn't think about that really from the compost standpoint, is that there could be some of these other materials that might be embedded in the feedstock, or that might be touching compost that perhaps needs some more additional explanation. So drill down a little bit in some of the kinds of PFAS that you looked at in the study that you did on PFAS. Did you all look at just a single PFAS? Were you looking at a suite of a bunch of different types of PFAS compounds? And can you help us understand that a little bit from the research that you've done?
Yes. So at the point when I was conducting the research, PFAS standard was available about 15 to 20. So we could see some important PFAS. I am saying PFAS, but majority of them were PFAA, perfluorinated alkyl acid. That is something close to PFOS, PFOS, or PFOA, PFOA. So in the slide, it's showing some brief structure of chemical. Based on their production mechanisms, we can say some are synthesized using fluorotelomerase method, and the other one is electrochemical fluorination. Most of the important and well-known PFAS are synthesized using electrochemical fluorinated method like PFOS and PFOA, and the other also important, but majority of them are precursor of PFOS or PFOA type compound like perfluoroalkyl acid compounds or fluorotelomers. So what I mainly looked at was perfluoroalkyl sulfonate and perfluoroalkyl carboxylate. And they are important because during the many degradation procedure, they ended up being this kind of PFOA or PFOS, PFSA, PFCA type compound, because they are more stable than their precursors and fluorotelomers. So I looked at total 13 PFAS compound in the compost.
Okay, so let me ask a follow-on question to that. So if I heard you correctly, the fluorotelomers are the base PFAS compounds that would be coming in from the feedstock, and then the PFSA and the PFCA, you're saying these are daughter products of the fluorotelomers that are forming as a result of the just as a natural breakdown of the parent PFAS molecule. Did I hear that correctly?
Oh, so PFSA and PFCA can exist as it is, because they can directly use for making products. But in case of fluorotelomers, it can be synthesized used for the products, but also can be transformed to PFSA or PFCA. So sometimes, although the feedstock has fluorotelomer, but after processing the composting procedure, what we may ended up detecting PFSA or PFCAs.
I see. And so this brings me to something I read in the paper you wrote, where there was a comment that a lot of the PFAS compounds detected were short chain PFAS. And so can you help us explain, or can you help the audience today understand a little bit about what do we mean by long chain and short chain, and why is the long chain short chain description important in terms of how we understand the impact of PFAS on composting?
Yeah, so based on the long chain PFC action plan by EPA, perfluorinated chemical action plan, they defined long chain PFAS is C8 perfluoroalkyl sulfonate and C6 sulfonate, that is perfluorooctanoic sulfonate, perfluorohexanoic sulfate, and their salt, and any precursor can be transformed to those chemical or longer chain of them, and perfluorooctanoic carboxylate, PFOA, or any longer chain PFCAs, and their precursors can transform to PFOA or longer chains. They were defined as long chain PFAS. That means there is something like 8 to 2 fluorotelomer compound they can transform to PFOA, so they are also long-chain PFAS. And in case of degradation of PFAS in compost, short chain does not want to use long chain for their food contact materials. It pushes the transition of chemical from longer chain to shorter chain. And 2015, PFOA was phased out its production and usage. So it's eventually changing the use of PFAS from longer chain to short chain. That's why I could see a lot of shorter chains than longer chain in the compost materials. And recently, FDA also claimed that the potential of harmful impact from 622-FTOH, which can be transformed to C6PFCA. So now, sometime later, we may not see even shorter chains because we don't want to even use that length of PFASs.
Okay, so the main reason you detected the short chain, just to summarize what you said, in compost was because of this historical shift from the, what we might say, the legacy C8, 8-chain carbon compounds to the shorter PFAS compounds being used in the feedstock materials. Is that right?
Yeah, that's correct.
Okay, and you mentioned food contact, and I know we really didn't hone in on this just yet. I did want to ask you, so when you were looking at the feedstock or at the compost, you mentioned food contact paper, and if I recall from the results of the paper, there was a pretty distinct difference between the compost that had contained food contact paper and compost that did not. Is that right?
Yeah, so when the compost contained food contact material, we could see much higher content of total PFAS, not just short chains, but so the total was even 5 to 12%, 20 times higher than the compost without food contact material. That means PFAS could reach it out of the food contact materials and increase the PFAS level in the final compost. And the upper side of the graph is showing PFAS content we detected. Majority of PFAS was shorter chains, and it was 70% or higher from the total PFAS we detected.
Okay, so mostly short chain, and it sounds like from what you're saying, mostly food contact paper would now, and I may be taking this into an area that you didn't explore, but did you all look at other types of compounds that might contain PFAS that would end up in feedstock for composting besides food contact paper? Were there any other types of things that could be coming in the feedstock that you would think may contain PFAS, or really is it the food contact paper is the primary culprit?
So for that compost that we speculated food contact material is the main source or the main source of PFAS, but at the same time, we tested other type of compost. Some was non-biosolid based product, and a couple of them were similar to food containing compost. And the most 10 of compost was biosolid based product, and we could see a lot of PFAS. The difference between the compost from municipal waste-derived compost and the other compost was we collected biosolid-based compost at 2014, but we collected food or municipal waste-based compost at 2017. So in the meantime, at 2015, we could know that there was volunteer phase out of PFOA. So since all the biosolid compost was sampled before 2015, we could see more longer chain in the sample. But when we collected same product in different time frame, 2016 and 2018, we could see the total PFAS was slightly decreased. Also, the level of longer chain was decreased as well. That means not only food contact material and food municipal waste solid containing compost, but overall the PFAS usage is changing, switched from using longer chain one to overall the shorter chain, whatever shorter than C8.
Okay. All right. In looking at the difference between a biosolid and non-biosolid, in terms of the types of compost. So, the non-biosolid compost would be, and this may sound obvious, but just for some of us who maybe still be on the learning curve with this. So for a compost that's non-biosolid, this is going to be a compost that would not contain biosolids from a wastewater treatment plant, right? And then for a biosolid base, this would be a compost that would have some type of biosolids from a wastewater treatment included. Is that correct?
Yeah, that's correct.
Okay. Okay, and then the profiles are definitely different, but still in either case there are PFAS compounds showing up. And is it fair, based on what you just said, again, just trying to summarize this for our audience, that for non-biosolid based composts, that the primary contributor is what you found is pretty much pointing towards this food contact as a source?
They are actually just yard trimming, yard trim waste, or or just food material without food contact materials, or something like mushroom waste, as we mentioned in the previous slide. So majority of them is considered there is really no PFAS input for the composting.
I see. So therefore, those concentrations are markedly lower then, since there's not a kind of a smoking gun of a source for PFAS there.
Yep.
Got it. Okay. Okay. So let's talk a little bit about. Well, let me ask you this: Do you feel the PFAS detected were essentially in the original form? I know we touched on that, but when you look at these 17 that were detected, are all of them pretty much going through the composting process with very little transformation, or are we seeing significant transformation of these 17 compounds as a result of the composting process into other types of PFAS, sort of daughter compounds? So, I mean, how resilient are the individual of the 17 PFAS that were detected? Like, how resilient are these to the composting process in being transformed, or are we seeing all of them, change in some form or fashion?
Yeah, so those 17 compounds, PFCAs and PFSA, they are almost impossible to transform by microbial activity. That means they are very final one, but there is a chance to have their precursors in the biosolid or their feedstock. So from composting procedure, the unknown precursor can transform to PFCA, PFSA, such as PFOA and PFOS. So this is why we call the PFAS precursors dark matter, because we have no information unless we really dig into and dive into to know what's in there. But as time goes by microbial activity, or when we do treatment technology applied to the PFAS contaminated sample, they can transform to the final very stable PFAS like PFSA and PFCA. So there are a couple ways we can measure the PFAS. So the total PFAS can be measured by counting, I would say count number of fluoride in the sample. And the most generous way, the common way we can detect is using standard chemical to know how much quantifiable PFAS is in the sample. And the other one is using high resolution mass spectrometry technologies. By using it, we cannot quantify them, but we can qualify them. So we can say we have some kind of chemical which can be transformed to PFSA or PFCA. And here in the paper, I use two different methods. One is using HRMS, high resolution mass spectrometry, to qualify the chemical can be transformed to PFSA or PFCA type chemical. And the other one was using TOP assay, total oxidizable precursor assay. By using this one, we can transform undetectable PFAS precursors to transform to quantifiable PFAS, such as PFSA and PFCA. When we use TOP assay, we cannot directly predict how much amount or level of precursors were in the sample. but we can briefly understand the rough amount of precursors in the original sample by measuring before and after the reaction. So after applying TAB-SA, we could see 2 to 5 times increased PFSA and PFCA concentration. That means there was chemical PFAS precursors could further transform to PFSA and PFCAs.
So, and talk to me a little bit. You're an analytical chemist, which is
Yep.
which means that you spend a lot of time with the methods in terms of how I interpret that. And we've heard in other areas of the PFAS discussion when you're talking about trying to detect PFAS in pretty much anything outside of sort of a clean source, such as drinking water, that detection of PFAS can be problematic. It can be very challenging. And looking at some of the equipment, the HRMS and some of the CIC, these aren't, this isn't standard stuff that I could, I couldn't just cook this up in my kitchen with a piece of litmus paper and figure this out, right? I mean, this is some pretty sophisticated chemistry to be able to make these detections in a quantifiable way. How challenging was this for developing the methodology that could find this? Is it is it a cakewalk, or is it something that took a fair amount of time to get the method squared away so that you could actually quantify this with some level of accuracy?
Yes. So one main reason is there's so much PFAS everywhere. That means while sampling and even extracting, cleaning, preparing sample, even during analysis procedure, the instrument or methanol, water, anything you're using could be already contaminated with PFAS. So that is one hard thing. It's eliminating base background PFAS. And the other thing is, since PFAS it's one group of man-made chemical, but it actually contains one group name. Under this name, there is more than 6,000 chemicals there. And it is already can be categorized by higher than 150 of classes, and PFSA, PFCA, or one of them. That means more than what we know, there's way more compounds are out there. So it is almost impossible to quantify individual chemicals. So that's why we're trying to understand by using instrument, we're trying to understand what's there. And once it is revealed, it's important to quantify. For example, PFOA and PFOS, they are pretty much, we know that they are harmful and they're very dominant compounds. So we are targeting and analyze them. Yeah, I think that is most of it. And then, yep.
So I do have a question. The testing that is done on total fluorine, do you have a sense for if somebody conducts a total fluorine test, like on any kind of environmental material, whether it's compost or whether it's landfill leachate or soil or what have you, how much of a total fluorine signature is going to be attributed to PFAS? In other words, how many compounds might exist out there that are not PFAS compounds that if you just did a total fluorine test, I kind of view that as we're casting a really wide net that kind of catches everything with fluorine, but I always kind of wonder what other fluorine compounds are out there that are not PFAS that we're not talking about right now that kind of show up in that kind of a test. And is the total fluorine test a reasonable test to say that when we see this fluorine concentration with a total fluorine test, that's mostly PFAS? I mean, but is that the right way to think about that? That a total fluorine is pretty much giving us mostly PFAS in that result, or is it that there are many other fluorinated compounds out there that are not PFAS that kind of add to the noise level when we look at the total fluorine number? Because I've seen other research that show total fluorine, and I feel like they make the assumption that if total fluorine is shown, then we're basically talking about PFAS, and I've not been able to really tease out from the literature whether that's a fair way to look at it or not.
I think that's a really good question. And so far, the total fluorine analysis never go it by itself, but always went with another technology to compare. So for example, there was always going with extract of fluoride analysis and total fluorine analysis, and HRMS analysis went together to compare how much they actually could obtain the recoveries. So as far as I know, many cases, the total fluorine analysis was supported for the AFFF tech analysis, because AFFF impacted site, majority of fluoride source could be PFAS. So it could successfully support the amount or the level prediction alongside the HRMS analysis. But if it goes with biosolid or compost, it would be a different story, because we know there are a lot of pharmaceuticals contain fluoride, not just PFAS. And in case of some studies about the biosolid, we could see there are 3 or 4 order of magnitude higher level of pharmaceuticals in biosolid that is way higher level than PFAS. So if we just do analysis of total fluorine to understand PFAS level, like the material, the matrix, such a complex material like compost and biosolid, that may not give us accurate analysis. So I believe we may want to do additional analysis such as HRMS analysis to qualify or quantify them. Also some other research like TAB assay, because it is only transforming some type of precursor to detectable, quantifiable PFAS. So it'll give us more reliable result to understand how much PFAS in there.
Okay. Now, so in this situation with the compost analysis, do you have a sense for what percentage of PFAS of those 17 compounds, what percent of those compounds were made up the total fluorine amount? Was it a small amount? Was it significant in this case, or were you able to analyze that?
So for the study I conducted, I didn't do the total fluorine analysis, so there is no direct comparison, but I could see there was huge amount of precursor was transformed to PFCA or PFSA after TAB assay. That means precursors were in the sample.
I see. Okay. Now, did you do any testing on any of the runoff or emissions from the compost operation, or was it just the compost directly?
I studied only compost directly, and I think there is no study conducted yet for the compost, but there are some study about landfill. So during the landfill procedure, there are some researches going on to understand how much PFAS is. PFAS could be evaporated as a volatile type, and they will give us more information about the composting procedure as well.
I see. Okay. Well, so once we have, with this work that you've done, we have an understanding that PFAS can certainly be in the compost. It seems to be more likely that it would be in compost that has had some kind of feedstock where food packaging has been a part of that feedstock. Once the composting has been completed and that product then is used, typically it would be land applied. So talk to us a little bit about the fate and transport in the environment once compost then is in use, whether that's in a field or in landscaping or elsewhere. What do we know about the fate and transport of PFAS, out in the environment as it relates to, PFAS and going from compost and then getting in soils or plants and so forth?
Yeah, so once compost is introduced to field, they can first leach out of the compost and soaked on the soil again. But where there is a precipitation, then they can go penetrate into the deeper part of the soil and also can go to groundwater. And another possible transport mechanism is runoff. The PFAS solved on the particle can go with water runoff. The other one is plant uptake. PFAS can be transformed via plant, and many studies showed shorter chain can transform more than longer chains. such as C4 and C5 PFCAs, PFBA, PFPEA. One study used lettuce and see how much PFAS was transformed from soil root to upper part. They found C4 and C5 detection from the lettuce, but when they tested with the corn, they couldn't not really detect meaningful amount from their corn, but they could detect some amount from their corn stalk. So what we also may want to know is what kind of crop will be more bioconcentrating or biotransporting from soil to their upper part. Also, the difference between the house farming and the field site would be the field farming can be impacted a lot by the climate and the more precipitation event, especially in case of Indiana. We are using a lot of pile drainage. Pile drainage also can change the flow of water. So we try to understand how the pile drainage changes the flow of water, and flow of water also can change reachability of PFAS. So that is another part we want to study.
Okay, so in addition to that, what other research is needed? I know this paper that you published, I guess it's dated 2019, which isn't really that old. I mean, we haven't seen any other research come out on the composting front. So what other kinds of research do you think is needed to help advance our understanding of how potentially to manage the composting side of things, to manage the feedstock, and then to consider what happens sort of downstream of this material?
Yeah, what the compost material, when we tested it, we saved it using 2mm sieve and tested the small particle parts. When we tested large part, the contribution to PFAS contribution to total compost was pretty low, less than 10%. That's why we more focused on the small particles. And although we focus on small particles, when it's mixed with large debris like wood, salt, or something, the total concentration per compost could be decreased. Also, when we follow the distribution or compost application instruction, then the application of PFAS from compost to the field site will be decreased more than even actual concentration we detected from the little particle. So that means when we mix with something with PFAS containing compost, that's the way we can dilute and decrease the PFAS level. As I mentioned before, PFSA and PFCA are very stable. There is no way to really break down in them in the compost, especially when we want to use compost, we cannot really break them. So there's the only way we can reduce is diluting them, and not just diluting them with peat or soil. There could be another good amendment we can add to reduce their reachability. So that is another part we may want to study.
Okay. Do you know, and so now with this, I know we've got about 15 minutes left, so let's move on to some of the attendee questions, because your last comment there reminded me of one question that we saw come in early on, and that's basically, is there anything that can be done to the feedstock in advance before it goes to a composting operation to help reduce the concentrations? And and one of the questions is can we use biochar, or can we somehow do a pre-treatment activity to try to reduce the concentrations? I think from a practical standpoint, one of the would be, if you're talking about food packaging, is to try to see how one can not have food packaging in that, whether that's through kind of acceptance criteria for some of the feedstocks, or some kind of physical separation process. But outside of that, as kind of an obvious takeaway, what are there any other strategies that you would point to that might be able to help reduce concentrations before in the feedstock before it gets to the composting facility?
Fortunately, other than the way you explained, not adding anything having possibility of high PFAS contents, not adding them, there's really no way you can reduce. But after finishing the composting procedure, we can add and dilute them. That's what we can reduce the level of PFAS from finished compost product.
Okay. All right.
And about the biochar, I know there is no direct research conducted with composting and biochar mixture, but there are a lot of studies showed from the PFAS removal using biochar from water, because biochar is really good at solving PFAS. And the other one is when biochar is mixed to PFAS contaminated soil, it helped reduce the reachability of PFAS. It was more effective for the longer chains and relatively less effective for shorter chain, because shorter chain has more reachability than longer chains. So when it's mixed with compost, it'll be really also helpful for reducing reachability of PFAS, also decreasing the concentration of PFAS from the total amount.
So that's great, thank you. And I have a lot of questions related to levels of PFAS, and so we'll try to hit all these together in sort of one class of questions here. One is in terms of just PFAS levels in compost. Is the perspective, and you might, I guess I'll say there's maybe a couple of different ways to look at this question. One would potentially be as a regulator, one would be as a chemist. But when we talk about PFAS levels, would any level of PFAS, no matter how small, be considered contamination, or is there a level at which, once we're below that, it's perhaps not considered contamination? And I realize we might be getting it really into a question that's more suited for a toxicologist to answer, but I don't know if you have any reaction or thoughts on that.
That's a good question, and as you expected, it'll be more good to give an answer with the research about the toxicology part. But what I know and what I can answer is usually longer chain is more toxic than shorter chain, as it's revealed so far. It's not super clear and super well defined sentence, because no one will really confirm 100% that shorter chain is always less toxic than longer chain. But in general, we believe longer chain is more toxic. So far, what I can say is longer chain and shorter chain may have different threshold of concentration. So for example, longer chain may have lower level of threshold than shorter chain, because they're more toxic and they're more hard to break down, and they're more dominant, especially of C8 and C6 PFAS. They're more dominant, so they're going to have lower level than shorter chains.
Oh, okay. Yeah, we've got a this other question really hits on, I think, the issue of comparability in different environmental mediums. To add syllables that probably I didn't need to add there in the question here. But so, for example, if we're trying to test, if we want to try to understand PFAS levels in drinking water, and we want to understand PFAS levels in compost, how would you compare those from as a regulatory agency, or even just as a facility owner? If I've got 30 parts per trillion in drinking water or in a groundwater situation, and I've got 20 micrograms of PFAS per gram of soil, or per how do you actually compare that to apples to apples? Is there a way to compare that in a reasonable way to get a sense for how I guess concentrated the PFAS may be in these different environmental sources?
Yeah, that's really difficult question, because we already know the answer that it's really hard to compare. So there are some approaches to understand the connection between the concentration in water or irrigation, water or compost, to the drinking water level or uptake per day level. There are some modelings about when the contaminated water is irrigated to the field, and they calculated how much will be transport from soil to plant, and they also calculated how much uptake as a produce consumption. So they did some calculation that this amount of contaminated water will end up this amount of intake. But this is still assumption, and it's good modeling, but still I can say it's a bit hard to connect directly the drinking water level to PFAS in compost, especially as we looked at the concentration of PFAS in compost and biosolid-based compost. The concentration is relative to the drinking water level. It already have pretty high concentration there. So I think we need to more focus on not just the concentration itself, but how we can eliminate them and how we can reduce them. Also, what would be the good process we can reduce the reachability of PFAS from compost.
Right, right. And I know we talked about this a little bit in the prior science session about, in some respects, the concentrations are helpful if you have a consistent exposure to that material. But if you've got, for example, an extraordinarily high concentration of PFAS in a material that you're really unlikely to get exposed to, that may not be as important as something that perhaps has a lower concentration that you might get routinely exposed to. So a lot of this does tie back to kind of the health implications, or I think we use as a surrogate for health implications the environmental contamination, because ultimately that's how things end up, in our bodies is through those different pathways. So that's a good point to note. It is very difficult to compare, and beyond just quantifying the levels, which I know is of prime importance, from a regulatory standpoint, because so many states and other agencies are looking at how to set these levels. And of course, for the composting industry and any other industry, that a lot of times has significant effects on operations, and when one has to, do testing or, adhere to some sort of threshold. So certainly, more research is needed to, I guess, better understand, some of these things. And we had a number of questions come in from the study you did, and maybe just to reiterate a little bit, the compost that you used in your research, can you contrast again the different types of compost that you tested and what was in that compost? In other words, where did it come from?
That is what we also wanted to know. But since it arrived as finished compost, it was hard to really track. But based on the information we received, we could know what was the potential input as a feedstock. So we didn't know what was really like all the amount or process everything, but we could assume what was impacted on the PFAS level in the finished compost.
Okay, so but in that respect, then you were able to quantify the difference of the different feedstocks having food packaging versus not food packaging, right?
Yeah.
Okay, great.
So we didn't know how much food contact material was added, but we could know it had it.
I see. Okay. And then just to clarify again, I mean, the composting process itself is not able to break down the precursors into more stable chains other than those, I think you said PFCAs, for example. Some of the parent compounds did turn into the PFCAs, but PFCAs also existed in the feedstock itself. Am I characterizing that correctly?
Yeah, so some precursor can transform to PFCA or PFSA, but they are already as it is, and they are stable, so they cannot be transformed to any other form in the composting procedure.
Okay, so in that respect, if somebody wanted to understand kind of a mass balance through a composting operation, could one test the feedstock at the beginning of the compost operation and test the finished compost and basically understand, based on the difference, understand whether anything was sort of lost throughout that process, or because of these transformations, would that be hard to do? I assume one could, I'm going back to our methodology discussion earlier, I assume one could do a total fluorine on the incoming feedstock and a total fluorine out, and that would still capture even any transformations that may have happened into some daughter products. Would that be one way to do that?
So in that case, we may want to do HRMS approach to know what we could see after or before the procedure, and we may also want to do total fluorine or other method to analyze total fluoride content before and after composting, because in many cases to analyze PFAS, we are using HRMS technology, but there are still a lot of PFAS we can analyze using these technologies. That's because it's more analytical chemistry part, but they are just simply cannot detected by the mass spectrometry, and sometimes they can be detected using GC, or some are just too big chemical, so they are out of the range of instrument can be detected. So, and those big chemical also can be transformed to small material, then that's what we can measure it. So for that case, I would say the total fluoride analysis and HRMS or other technologies to measure its PFAS qualitative and quantitative analysis should go together to compare before and after the PFAS, the composting procedure, to see how much PFAS is impacted and transport.
So you mentioned detection limits. That's a great segue into one of the questions we had. What are the detection limits for the the HRMS technologies or some of these other methods relative to the what you detected in the compost? Were they close to those detection limits or high above? So what would be the minimum level one could detect using the methods that you used?
So the instrument itself can detect to part per trillion level, but we are doing extract and concentrate the PFAS from the initial material. So the actual concentration we can detect could even get lower because we are concentrating it. So,
okay.
Yeah.
And that's a pretty common practice, isn't it? If you have low concentrations in whatever sample material, is to basically get more sample and concentrate that versus something that's highly concentrated prior to running the test. You would then dilute that. That's relatively common practice in an analytical lab, is it not?
Yes, and there's many technologies we're trying to apply and develop. So for example, when we extracted sand compost a couple of years ago, several years ago, and now we can detect more PFAS because our extraction method modified and our instrument is also updated and our analysis method is optimized. Also, we have more PFAS standard we can confirm its existence. So before, more than several years ago, now we can do better analysis, and also we can do we can have better understanding about PFAS in the sample.
Mm-hmm. Now, are you aware of anything that's happening on the regulatory side of things with the EPA or state agencies about any potential regulations on or regulation limits on PFAS in compost at this point?
For now, I don't have any, but I know we are doing a lot of researches to understand the PFAS transport and transformation when it is applied to the field site, and that I expect they will give us information of how much we need to have a level of concentration for the legislatively or can decide for the company.
Gotcha. Okay. Well, I think that brings us to the top of the hour, and I know we have a number of other questions. We will try to send those over to you, Dr. Choi, and if you're able and willing to provide a few more items of input, we'll then circle that back out to folks as best we can. But I want to thank you very much for joining us today. Thanks for giving us a bit more insight. Very helpful. I also got a sense that there's a lot more that we can do to better understand what's happening in compost on PFAS, but this initial work that you've done has been extraordinarily informative in helping those of us who are tracking the science and trying to understand what's happening and how all these pieces fit together. So we appreciate that work you've done. And are you continuing to do any work on this front? And if so, like what is your lab doing next?
So what we're doing is we are studying more about biosolid based compost and distribution of biosolid to the field site. What we are trying to know is what is good amendment and application procedure to see how PFAS can be released and go to the field site. So this is what we are doing, and we are also trying to understand what kind of composting or biosolid treatment part can change the PFAS level, and the type of biosolid can impact on the PFAS leachability, etc. So we are trying to know about it now.
Okay, great. Well, again, thank you for that. Thank you for your time today. And for those of you who joined us, we thank you for your time. And I hope you'll join us as part of the next science sessions, or in between the next science sessions. We'll hope you will join us for the training session. Thank you, everyone. Thank you, Dr. Choi. And I hope everyone has a great day and a great rest of the week. Take care.
Thank you for having me. Thank you.
Mm-hmm.